Good morning, everyone, and welcome to today's Journal Club. Today, I'll be presenting a groundbreaking paper from Nature on AlphaFold3, which represents a revolutionary leap forward in our ability to predict the structure of biomolecular interactions. This work, led by Josh Abramson and colleagues at DeepMind, extends far beyond single protein prediction to tackle the challenge of modeling entire molecular complexes. Now, let's set the stage by acknowledging the revolutionary impact of AlphaFold2 on protein structure prediction. While AF2 achieved remarkable success for individual proteins, the fundamental challenge in molecular biology remains understanding the complex atomic interactions that govern biological systems. This requires moving beyond single proteins to a unified framework capable of predicting diverse biomolecular complexes, including nucleic acids, small molecules, ions, and modified residues. Now let's examine the key architectural innovations in AlphaFold3. The most significant change is the adoption of a diffusion-based approach that completely replaces AlphaFold2's structure module, allowing for more flexible structure generation. Additionally, the new pairformer module streamlines the computationally intensive multiple sequence alignment processing, while the system now operates directly on atom coordinates rather than being constrained to amino acid-specific
오늘 저널 클럽에서
reference frames.
소개드릴 논문은 Nature
Moving to the training methodology, AlphaFold3 introduces several key innovations to improve prediction accuracy. The cross-distillation approach significantly reduces hallucination artifacts that plagued earlier models, while a novel diffusion rollout procedure allows the system to predict complete molecular structures. The training follows a hierarchical strategy where the model first masters local structural features before progressing to complex interchain interactions through staged training with progressively larger crop sizes. Moving to protein-ligand interactions, AlphaFold3 demonstrates remarkable docking performance that significantly surpasses current state-of-the-art methods. The model shows substantial improvements over specialized docking tools like Vena and Gold, achieving accurate predictions without requiring any prior knowledge of binding pocket locations. This blind docking capability is particularly impressive when we look at challenging cases where traditional methods have failed, such as the human NOTAM inhibitor complex shown here, with an RMSD of just 0.65 angstroms. Moving to protein nucleic acid complexes, AlphaFold3 demonstrates remarkable capabilities in predicting these challenging systems. The model significantly outperforms specialized nucleic acid predictors like Rose's TTFold2NA and can handle massive complexes with thousands of residues. This represents a major advance in our ability to model the intricate interactions between proteins and nucleic acids at unprecedented scale. Moving on to one of the most impressive applications, AlphaFold3 shows remarkable performance in antibody-antigen prediction. The model achieves substantially higher accuracy in predicting how antibodies bind to their targets, with marked improvements over previous specialized methods that were designed specifically for immune system interactions. What's particularly noteworthy is that this unified framework handles the full diversity of immune complexes without requiring any task-specific modifications, representing a significant methodological advance. Moving on to one of the most impressive capabilities of AlphaFold3, it can predict covalent modifications and modified residues with remarkable accuracy. The model handles everything from bonded ligands and glycosylation to post-translational modifications of proteins and modified nucleic acid bases. This represents a major advance beyond traditional structure prediction, as the evaluation methodology carefully separates bonded from non-bonded ligands to assess these complex interactions properly. Moving to our validation results, we conducted extensive benchmarking using interface-specific datasets derived from recent PDB structures. Our evaluation methodology was rigorous. We excluded crystallization aids, applied homology filtering, and used clustering approaches to ensure statistical robustness. The model demonstrates strong generalization across diverse molecular interactions, from protein complexes to nucleic acid binding and small molecule recognition, with separate validation for different interaction types. In summary, AlphaFold3 represents a transformative advance in computational biology through its unified diffusion-based framework that can predict structures across diverse biomolecular systems. The model consistently outperforms specialized tools while demonstrating remarkable generalization capabilities, even without cross-entity evolutionary information. This work takes us substantially closer to the ultimate goal of understanding and regulating the complex atomic interactions that drive biological processes.